Antiviral drugs. So methodology of antiviral drugs. First of all, you should know what do you mean by virus. So all the microorganisms like bacteria, fungi, okay, rickettsia. So virus is also a microorganism. It is also a foreign microorganism for your body. But the major difference between bacteria and virus. What is the major difference? The major difference between bacteria and virus is the virus is intracellular parasite. Bacteria is not an intracellular parasite. Bacteria will have its own cellular structure. Bacteria will have its own in the cell also, or it can live without the cell. But virus is an intracellular parasite. That means you cannot isolate virus as it is. So if you want to isolate the virus, then you have to isolate it from host cell only. कि देखो ये जो फॉरेन जो वायरस होता है, जो आपका सेल होता है, आपका सेल मतलब your body cell means host cell. Okay, so virus are the intracellular parasite. It can virus lives only in the intracellular. It is dependent on the host cell for its metabolism, for its energy, and for its each and everything, for its replication, for and every kind of function. Okay, for the survival. So virus is an intracellular parasite and totally depends on the host cell growth, multiplication, and all that. It cannot grow or multiply by itself, so it requires host cell. So you cannot isolate virus as it is. So it can be only isolated in the infected cells only. अगर आपके पास infected cells है वायरस का, तभी आप उसको isolate कर सकते हो. And again, you cannot grow the virus on non-cellular medium. You cannot grow virus on the culture medium. So for the culture medium, you need cells. Okay, you need a cell. So virus is an intracellular parasite, and that depends totally on the host cell for growth and multiplication. Okay, now there are different steps involved in the growth of bacteria. There are different steps involved in the multiplication of bacteria. These are the steps. For example, first viral particle or virus will attach to a host cell. Okay, so virus will first attach to a host cell. So जो भी host cell है आपका, so whatever host cell is there, so virus will first attach to a host cell. Then only it can, then after that it can penetrate into cell. After attachment, virus will penetrate into cell. It will go inside the cell. So it will attach with the cell via different kind of receptors, via different kind of सेलुलर रिसेप्टर्स. सेल के ऊपर जो मेम्ब्रेन होता है, सेलुलर सेल मेम्ब्रेन, उसके ऊपर डिफरेंट काइंड के ग्लाइकोप्रोटीन्स होते हैं. So there are different glycoproteins which are present on the host cell, and these glycoproteins are the receptor for virus. Okay, so virus will attach first to the cell via these receptors. We'll discuss the which kind of receptors are these. We'll discuss in the next we'll discuss in the upcoming slides. Okay, so virus will attach to a host cell, then it will penetrate into the cytosol of your cell. Okay, then attachment of virus to host cell membrane, then penetration of virus in the host cell. Attachment के बाद virus अंदर जाएगा सेल के अंदर, so it will penetrate in the cell, then it will uncoat himself. जो भी उसका कैप्सिड का स्ट्रक्चर होता है, कैप्सिड के स्ट्रक्चर में वायरस के अंदर, so whatever capsid is there of virus, inside the capsid there is genetic material of virus, whether it will be either DNA or RNA. It depends on the type of virus. So there are two types of virus, DNA virus and RNA virus. Okay, so अगर वो DNA virus है तो DNA release करेगा, अगर वो RNA virus है तो that will release RNA. So after penetration into host cell, it will undergo uncoating. Okay, so whatever coating is there on the capsid, whatever coat of capsid is there, it will undergo uncoating and release of viral genetic material. Okay, now the genetic material is released in the cytosol of human cell or host cell. Okay, now that genetic material is released along with the few enzymes also. This DNA or RNA of virus will get multiplied or will get replicated in the host cell. Okay, so in the cytosol by using host DNA polymerase. होस्ट डीएनए पॉलीमरेज, आपका सेल का जो डीएनए पॉलीमरेज एंजाइम है वो वायरस यूटिलाइज करेगा उसके डीएनए के, उसके खुद के डीएनए के या फिर आरएनए को मल्टीप्लाई करने के लिए. Okay, so virus will uncoat himself in the cell of host and then it will release its genetic material. Then this genetic material is replicated by using host enzyme like DNA polymerase. Okay, So fourth step will be the synthesis of viral component. So the component tail and capsid, the protein responsible formation of capsid and the protein responsible formation of tail, those will be synthesized in a different steps, and again a genetic material of virus will be synthesized in another step. Okay, and all these are assembled in the host cell itself. Okay, after that, after the assembling of DNA or DNA, after the assembling of genetic material, capsid or and tail, then the virus will get mature. Okay, with the help of its enzymes, and then it will be released from the cell, and it is ready to affect the another host cell. So that's how virus will live in our cell. So virus is an intracellular parasite. It totally depends on the host cell for its growth and multiplication. So normally, which are the steps involved in the growth or viral multiplication in the host cell? Host cell मतलब क्या है? आपका cell. Okay, so host cell मतलब human cell or any other animal cell wherever there is infection. That is a host cell. First of all, virus will get attached to a host cell membrane by using various kind of receptors, glycoprotein receptors. Then second, after attachment, the virus will get penetrate into the cell. Okay, so after crossing the cell membrane, it will go into the cytosol. There the virus will get uncoated itself and release the its genetic material in the cytosol. That is the step 3. Now after this, the genetic material of virus get replicated. Okay, now the replication will be there, then transcription will be there, then translation will be there. Okay, so replication means if DNA virus is there, there is a replication. If RNA virus is there, then first RNA is converted into DNA and then it is replicated, then it is transcripted, and then it is translated. Okay, so this now step 4 is the synthesis of viral component. Viral component means capsid and tail. Whatever protein of capsid is there, whatever protein of tail is there, so there these proteins are synthesized separately. Then genetic material of virus is uh, preserved separately, and all these three are assembled, and virus is matured. Okay, by process of maturation. Then this there will be release of virus from your host cell. Okay, so infected host cell, so there will be release of many viruses. Those are ready to affect the another cells. Okay, so these are the this is the just overview of life cycle of your virus. Now antiviral drugs, they are mostly non-selective in nature, unlike your antibacterial drug. So antibacterial drug में क्या होता है कि they are selective in nature. So whatever antibacterial drugs are, जैसे सल्फोनामाइड्स हो गए, आपका पेनिसिलिन हो गया, aminoglycosides. So antibacterial drug, they are selective in nature because so there is a little bit difference between microbial cell and human cell. That's why antimicrobial drug, they are selective in nature, but there is no difference between there is no difference between virus and human cell. Because why? Because virus itself is living in the cell, so you cannot destroy your cell selectively. So antiviral drug are non-selective in action and can affect the host cell also. That's why antiviral drug also have multiple adverse effect. Okay, so antiviral drug, because virus is living in the cell only, virus is living in the host cell. Host cell means human cell. Okay, that's why uh, virus is also utilizing your host cell. आपका जो होस्ट सेल है, आपका जो सेलुलर एंजाइम्स है, सेलुलर एंजाइम्स जैसे डीएनए पॉलीमरेज है आपका खुद का, वही यूटिलाइज करेगा आपके। जैसे सेल में आपका सेल की साइटोसोल में आपका प्यूरिन्स है, पिरिमिडिन्स है ना, प्यूरिन्स विटामिन बेसिस। सो दो वो बेसिस लेके वो आपका खुद का मटेरियल बनाएगा। मतलब उसका खुद का कुछ नहीं होता उसके पास, सिर्फ उसका एक बार जेनेटिक मटेरियल पहले जो जेनेटिक मटेरियल है वही होता है उसका। बाद में एंजाइम्स वगैरह जो भी जेनेटिक एंजाइम्स लगेगा, डीएनए पॉलीमरेज वगैरह, उसके बाद जो भी न्यूट्रिशन लगेगा, जो भी base pair लगे, nucleoside base pairs, so that will be utilized from host cell itself. So that's why you cannot selectively kill the virus. So but there are some targets, there are different targets for antiviral drug. Okay, so for example, we have discussed the 5 steps, that is attachment, penetration, uncoating, then replication, then synthesis, then synthesis of protein, viral component, assembling and maturation and release. So drug can affect at any one of the above stage or any combination of above stages. So जो 5 steps है वायरस के मल्टीप्लिकेशन के, in the bacteria in the host cell. ठीक है, host cell में भी either human cell or either animal cell or either microbial cell, कि microbial cell के ऊपर भी वायरस अटैक करता है, ऐसा नहीं कि वायरस सिर्फ ह्यूमंस को अटैक करता है. ओके, सो drugs can affect the viral multiplication, or drug can affect the virus at any one of the above 5 steps or any combination of steps. Okay, so there are two kinds of virus mostly, DNA virus and RNA virus. So depending upon their genetic material, कि वायरस का जेनेटिक मटेरियल क्या है. अगर उसका डीएनए है तो उसको बोलेंगे डीएनए वायरस, अगर उसका आरएनए है तो उसको बोलेंगे आरएनए वायरस. ओके, सो डीएनए वायरस एंड आरएनए वायरस आर द two major types. Okay, so the major difference in two type of virus, that is genetic material. So in the DNA virus you will have DNA as a genetic material. In case of RNA virus, RNA will be as a genetic material. Okay, and there is another difference. So DNA virus and RNA virus, besides having the different genetic material, they will have different replicative cycles. So DNA virus will have different replicative cycle, RNA virus will have different replicative cycle. DNA virus mostly depends on the DNA virus depends on the host cell enzyme mRNA polymerase for the synthesis of mRNA from DNA. But RNA virus will have its own mRNA polymerase mRNA polymerase for synthesis of mRNA. क्या होता है कि डीएनए वायरस आरएनए वायरस क्या डिफरेंस है. डीएनए वायरस में आपका डीएनए जेनेटिक मटेरियल होगा, आरएनए वायरस में आपका आरएनए जेनेटिक मटेरियल होगा. ठीक है, और एक डिफरेंस है उनका जो रेप्लिकेशन जो साइकिल है उसमें भी डिफरेंस होता है. जैसे डीएनए वायरस में क्या है, डीएनए वायरस विल यूटिलाइज होस्ट एमआरएनए पॉलीमरेज. ठीक है, डीएनए वायरस क्या करेगा कि जो होस्ट सेल का जो आरएनए पॉलीमरेज है एमआरएनए पॉलीमरेज वो यूटिलाइज करके एमआरएनए बनाएगा. ठीक है, और जो आरआरएनए मतलब आरएनए वायरस जो होता है वो उसके पास खुद का एमआरएनए पॉलीमरेज एंजाइम होता है. ठीक है, so that is the major difference between two type of viruses. There is another virus known as rotavirus. Sorry, another virus known as a retrovirus. Retrovirus. So retrovirus is a type of RNA virus. Retrovirus is a type of RNA virus, and because of this immunodeficiency virus, human immunodeficiency virus, that is responsible for the AIDS, cause division for the AIDS. जो HIV वायरस होता है उसकी वजह से एड्स होता है. So that is a type of retrovirus, and it will have one extra enzyme known as a reverse transcriptase. It will have a reverse transcriptase enzyme. So what is the role of reverse transcriptase? नॉर्मली क्या होता है कि आपका जो डीएनए है, डीएनए से एमआरएनए बनता है आपका, और एमआरएनए से बनता है आपका प्रोटीन. एमआरएनए से बनता है आपका प्रोटीन. पहले क्या होगा कि डीएनए का रेप्लिकेशन होगा, डीएनए के भरपूर कॉपीज बनेंगे, उसके बाद डीएनए से बनेगा एमआरएनए, उसके बाद एमआरएनए से बनेगा प्रोटीन. Replication of DNA, making a multiple copies of DNA, it is known as a replication, and making of mRNA from DNA, it is known as a transcription, and making of protein or synthesis of protein from mRNA, it is known as a transcription. So we have seen in the biochemistry. But in case of retrovirus, the genetic material itself is a RNA virus. RNA. So retrovirus में RNA genetic material होता है. So first of all, he has to synthesize the DNA from RNA. For that, रिवर्स ट्रांसक्रिप्टेशन मीन्स उल्टा ट्रांसक्रिप्शन होने के जो होता है ट्रांसक्रिप्शन उसके उल्टा. सो दैट इज नोन एज अ रिवर्स ट्रांसक्रिप्टेज. रिवर्स ट्रांसक्रिप्टेज इज द एंजाइम that can make a DNA from RNA, and that is present in the retrovirus, or that is known as immunodeficiency virus, HIV, human immunodeficiency virus. So it is a type of retrovirus. So it will have a reverse transcriptase enzyme responsible for the replication of RNA. First RNA will be converted to DNA, and then it will be replicated, and then again mRNA and protein synthesis. Okay. First of all, the whatever viral proteins are there, they are immature viral particle or immature or inactive viral. They are formed in the cell, and after that it becomes mature with the help of an enzyme known as a protease. Okay. So क्या होता है कि नॉर्मली जब वायरस इन्फेक्शन करता है सेल को, तो वो अटैचमेंट होने के बाद वो अपना अंदर जाएगा, इनसाइड द सेल पेनिट्रेशन होगा, उसके बाद में रिलीज करेगा जेनेटिक मटेरियल. तो जेनेटिक मटेरियल उसका जो भी है उसका रेप्लिकेशन हो जाएगा. जैसे जेनेटिक मटेरियल डीएनए है तो उसका रेप्लिकेशन हो जाएगा, उसके बाद में ट्रांसक्रिप्शन हो जाएगा, उसके बाद में ट्रांसलेशन मीन्स प्रोटीन सिंथेसिस. और प्रोटीन कौन सा सिंथेसिस होगा, वो वायरस का होगा, अपना नहीं. जो वायरस के सेल कैप्सिड के लिए जो लगता है, टेल के लिए जो लगता है, वो प्रोटीन सिंथेसिस होगा. और बाद में वो प्रोटीन जो सिंथेसिस होता है दैट इज नोन एज अ that is known as that is known as a large protein, large inactive protein. And after that, it is broken down and it is activated by one enzyme known as a protease. जैसे protease उसको activate करेगा तो उसको बोलेंगे functional protein. And that protein is utilized for the making of parts of virus. And after that, there will be assembling of virus and release of virus. Okay, this is just a overview. Now next one is examples of DNA and RNA virus. Okay, examples क्या है उसके? Examples of DNA and RNA virus. So DNA virus, for example, herpes simplex, varicella zoster, smallpox, hepatitis B, human cytomegalovirus. So these are the DNA virus. So herpes simplex will have DNA as a genetic material. Varicella zoster, smallpox, hepatitis B, human cytomegalovirus will have DNA as a genetic material. RNA virus, for example, rabies. आपका रेबीज का इंजेक्शन, कुत्ता ने काट लिया आपको एक रेबीज का इंजेक्शन देते हैं आपको. रेबीज इज बिकॉज ऑफ द दैट रेबीज वायरस. सो इट इज आरएनए वायरस. रुबेला, measles, dengue virus, yellow fever virus, then poliomyelitis virus, HIV virus is there, and influenza virus is there. So these are the RNA virus. तो HIV वायरस में क्या है? RNA जेनेटिक मटेरियल है. पोलियो वायरस जो है उसमें भी RNA. इन्फ्लुएंजा वायरस जो है, सो इन्फ्लुएंजा ए, इन्फ्लुएंजा बी. सो वट एवर द कोरोना वायरस इज देयर. आजकल जो कोरोना वायरस का पैंडेमिक चल रहा है, तो कोरोना वायरस इज नथिंग बट द टाइप ऑफ इन्फ्लुएंजा वायरस. इट इज अ टाइप ऑफ इन्फ्लुएंजा वायरस. ओके. Then dengue virus is also there. Dengue virus is known as a flavivirus. So that flavivirus also has a RNA as a genetic material. Okay. Now this is the diagram demonstrating the different sites for action of antiviral drug. Suppose that this is your virus particle. ये जो पिंक में है ये वायरस है. उसका जो हेक्सागोनल स्ट्रक्चर है ऊपर का उसको बोलते हैं कैप्सिड. और ये जो बाकी नीचे वाला है उसको बोलते हैं टेल. नाउ कैप्सिड एंड टेल. दिस इज अ वायरस. वायरस विल गेट अटैच्ड टू योर होस्ट सेल. दिस इज योर होस्ट सेल. आपका सेल जो है ये है. ठीक है. वायरस विल गेट अटैच्ड, देन इट विल गेट पेनिट्रेटेड इनसाइड द सेल. इट इज नथिंग बट द cytosol. Okay. After penetration it will get uncoating. Uncoating means ये dissociate हो जाएगा और genetic material जो है उसका वो release करेगा. DNA RNA जो भी है. After releasing the genetic material will be there will be replication, there will be transcription, there will be translation. Okay. And after translation, protein synthesis will be there. So these proteins are inactive proteins. Protease enzyme will activate them and it will convert into, it will slice them, it will cut down. Okay. जो पहले प्रोटीन बनता है वो इनएक्टिव प्रोटीन होता है और लार्ज प्रोटीन बनता है. बाद में उसको स्लाइसेस बनेगी बाय यूजिंग प्रोटीज एंजाइम. ये प्रोटीज एंजाइम जो होता है वो वायरस के इसमें होता है प्रोटीन. नाउ फॉर द रेप्लिकेशन जो हो रहा है यहाँ से डीएनए आरएनए का जो रेप्लिकेशन, ट्रांसक्रिप्शन, ट्रांसलेशन. So for all three process, replication, transcription, translation, so virus is using host enzymes. जो ह्यूमन सेल का एंजाइम होता है जैसे डीएनए पॉलीमरेज, ह्यूमन डीएनए पॉलीमरेज, ह्यूमन एमआरएनए पॉलीमरेज, ह्यूमन प्रोटीन सिंथेसिस का जो है. सो वट एवर एंजाइम्स रिक्वायर्ड फॉर द रेप्लिकेशन, ट्रांसक्रिप्शन, ट्रांसलेशन of the virus, the virus does not have its own enzymes. It is utilizing your host cell enzymes. That's why it is an intracellular parasite, and that's why you cannot selectively kill the virus. After the synthesis of protein, so it will be broken down into active part, like this part will be found, like capsid will be there and tail will be there. Okay, and then this will be assembled, and then final virus will be ready. The maturation will be there. So assembly or maturation, and then virus will get released from the cell, and now this virus will release, it can affect another cell. So this is just a life cycle of virus inside the cell. Okay. Now during the attachment, so different kind of receptors are there responsible for the attachment, like glycoprotein, chemokinin receptors, etc, etc. At the attachment step only, so these kind of drug will affect. For example, n-flu-viridine, then miravirus, pelicillizumab. Okay. So these drugs will act on this stage. So they will prevent the attachment of virus to the bacterial cell, attachment of virus to the host cell. So n-flu-viridine, then miravirus, pelicillizumab. So this is the monoclonal antibody. This will prevent the attachment of virus to the cell, host cell. Now after attachment it will get penetration. अगर यहाँ पे आप ब्लॉक नहीं कर सकते हो, ठीक है, चलो पेनिट्रेशन हो गया, यहाँ पे अंदर आया. So it will get uncoated means it will release the DNA, it will release the genetic material. Okay. So during this stage, these two drugs, amantadine and rimantadine, these drugs can prevent the uncoating of viral DNA or viral genetic material. This amantadine also prevent the attachment also. The amantadine has a multiple site of action on the virus. Amantadine will prevent the attachment also. अगर यहाँ पे नहीं ब्लॉक कर सकते तो ठीक है, अनकोटिंग हो गया, अभी जेनेटिक मटेरियल रिलीज हो गया उस सेल में. So that genetic material, whatever it is, DNA or RNA, so it will be converted into, it will be replicated. Okay, it will be replicated. Replication will be there. Transcription will be there. Translation will be there. Okay. So for example, just take example of replication. So for the replication, DNA polymerase enzyme is important. Okay. So DNA polymerase enzyme is required. So there are the drugs which can inhibit the DNA polymerase enzyme. So DNA polymerase inhibitor will act on this stage. Okay. And then after that, after that transcription will be there, then translation will be there. Translation means protein synthesis. So protein synthesis में क्या होता है, during protein synthesis, so your जो भी आपका nucleosides होता है सेल में, जैसे adenine, guanine, thymine sites, so they are incorporated, okay. So they will code and then they will mRNAs, they will code the mRNA, okay. So these for the transcription, so these nucleotides at this stage, various kind of drug like this nucleotide reverse transcriptase inhibitor, non-nucleotide reverse transcriptase inhibitor, they will act on a RNA virus. NRTIJ, nucleotide reverse transcriptase inhibitor. So reverse transcriptase enzyme किसमें होता है, जो RNA virus होता है उसमें होता है. Reverse transcriptase inhibitors and non-nucleotide reverse transcriptase inhibitor. So these drug will also affect on the this stage during the transcription on the RNA virus. Now, if protein is synthesized, then there are few drugs which are known as a protease inhibitors. Protease inhibitors. So they will act on this stage. They will act on this stage. So they will inhibit the protease and they will prevent the activation of the protein. They will prevent the slicing of protein. And if you cannot prevent this stage, you can prevent this stage. You can stop the assembling of virus. Okay. So assembling of virus is stopped by rifampin. Then again release. So virus is released from the host cell by using neuraminidase enzyme. So this is again a target for antiviral drugs. So few drug they are known as neuraminidase inhibitors. So this is how you can classify the drug. So attachment inhibitors, then uncoating inhibitors, then DNA polymerase inhibitors, nucleotide reverse transcriptase inhibitor, non-nucleotide reverse transcriptase inhibitors, then protease inhibitors. Okay, then attachment inhibitor. Assembly, they will inhibit assembling inhibitors or budding inhibitors, then release inhibitors. So these are the classes. These are nothing but the targets, and these are nothing but the classes of drug. So that is nothing but the sites for antiviral drugs. Okay, sites for antiviral drugs. Now we'll see the classification of antiviral drug. Classification of antiviral drugs. First, class and example. So these antiviral drugs are classified into three types. Okay. So first one is a non-retroviral antiviral drug. Non-retroviral antiviral drug. नॉन रिट्रोवायरस मतलब रेट्रोवायरस नहीं है, मतलब डीएनए वायरस. ओके. सो नॉन रिट्रोवायरस मतलब non RNA virus. So non-retroviral drug means they are useful in the other viral infection, not in the HIV. Okay. So non-retroviral antiviral drug, for example, those drugs which are used against the herpes zoster. So anti-herpes virus drug or anti-herpes viral drug. Okay. So example is iodoxuridine, then this, okay, iodoxuridine, trifluridine, acyclovir, vanciclovir, ganciclovir, cidofovir, and foscarnet. So iodoxuridine, trifluridine, then acyclovir, vanciclovir, ganciclovir, cidofovir, and foscarnet. These are the anti-herpes virus. So they are used against the herpes zoster virus. Okay. Then next class is anti-influenza drug or anti-influenza antiviral agent. So those are effective against only influenza virus, okay, mostly. So for example, amantadine, rimantadine, oseltamivir, and peramivir. Amantadine, rimantadine, okay, oseltamivir, and this peramivir. These are the anti-influenza drug. There are a lot of examples, other examples also. Okay, I have mentioned some few examples only. Now third class will be their drug used in the hepatitis. So hepatitis means inflammation of your liver. So there are two types of hepatitis. So bacterial hepatitis and viral hepatitis. So virus causing hepatitis known as viral hepatitis. So hepatitis because of viral infection, that is known as viral hepatitis. And there are three virus. There are three virus which causes the hepatitis. Hepatitis A virus, hepatitis B virus, and hepatitis C virus. But hepatitis A is not a virus. So hepatitis A is not given over here. So whatever viral hepatitis are there, it is mostly of two: hepatitis B and hepatitis C. So hepatitis B virus is there and hepatitis C virus is there. Okay. So though those two viruses are causing the viral hepatitis. Okay. So hepatitis B, against the hepatitis B. So drug used against the hepatitis B virus, which are the lamivudine, then entecavir, adapavir, lamivudine, entecavir, adapavir, dipivoxil, then tenofovir, and telbivudine. So lamivudine, entecavir, adapavir, dipivoxil, tenofovir, and telbivudine. These are the drugs against the hepatitis B. Now, drugs used against the hepatitis C. So, example is ribavirin, interferon alpha. Interferon means viral antibody. जैसे होता है ना बैक्टीरियल इन्फेक्शन हो गया आपको तो उसके अगेंस्ट में एंटीबॉडी बनती है बॉडी में. ठीक है. जैसे वायरल का इन्फेक्शन हो गया वायरस का तो उसके अगेंस्ट भी एंटीबॉडी बनता है आपका, उसको बोलते हैं इंटरफेरॉन. इसके बारे में डिटेल में बाद में देखेंगे. सो रिबाविरिन, इंटरफेरॉन अल्फा, then soposbuvir, then simipravir, then decaltavir, then ledipavir, then vepatavir. So these are the drug used against the hepatitis C. So this anti-herpes drug, anti-influenza drug, and drug anti-hepatitis antiviral drug. So they are the non-retroviral antiviral drug. ये retrovirus नहीं होगा, ये DNA virus होगा. Drug used against the retroviral. So anti-retroviral drug. Next one is a anti-retroviral drug. In case of anti-retroviral, so retrovirus मतलब क्या है? उसमें reverse transcriptase होगा, उसमें RNA genetic material होगा, ठीक है? RNA as a genetic material होगा. तो retrovirus is a kind of virus which has a reverse transcriptase enzyme that will convert RNA into DNA, and then DNA will be replicated. Then again synthesis of mRNA and then protein synthesis. So these are the retrovirus. So against the retrovirus or against the anti-HIV drug, you can say it as anti-HIV drug. So against the retrovirus or drug used in the HIV. So NRTI, nucleoside reverse transcriptase inhibitor. न्यूक्लियोसाइड, तो ये क्या होगा? न्यूक्लियोसाइड का डेरिवेटिव होगा, जैसे एडेनिन का, थाइमिन का, ग्वानिन का. ठीक है. सो थाइमिडीन, थाइमिडेट, ग्वानिन, लाइक दिस. So they are the derivative of nucleoside. Okay, substituted derivative of nucleoside. So nucleoside reverse transcriptase inhibitor. For example, zidovudine, didanosine, then stavudine, lamivudine, abacavir, and tenofovir. Okay, so these are the नॉन रिवर्स ट्रांसक्रिप्टेज इनहिबिटर. इसके ऊपर क्वेश्चन आता है कि कौन सा रिवर्स ट्रांसक्रिप्टेज है, कौन सा नॉन रिवर्स ट्रांसक्रिप्टेज इनहिबिटर है. सो यू शुड बी क्लियर. सो दिस जिडोवुडीन, जो डी आयन है, वुडीन जो होते हैं, वुडीन, जिडोवुडीन, स्टावुडीन, लैमिवुडीन, ठीक है, डिटानोसाइन, टेनोफोविर एंड अबाकविर. दो ही है इसमें, अबाकविर, टेनोफोविर. ये है आपका न्यूक्लियोटाइड रिवर्स ट्रांसक्रिप्टेज इनहिबिटर. सो वुडीन एंड वीर आर nucleotide reverse transcriptase inhibitor. Now rapines, DB-rapine, ridine and rapines, okay, nevirapine and all that. So non-nucleotide reverse transcriptase inhibitor. अब ये न्यूक्लियोटाइड है, इसमें न्यूक्लियोटाइड रहता है, ठीक है, मतलब एडेनिन, प्यूरिन, ग्वानिन. सो नॉन न्यूक्लियोटाइड रिवर्स ट्रांसक्रिप्ट इनहिबिटर मींस इसमें न्यूक्लियोटाइड का स्ट्रक्चर नहीं रहेगा. ठीक है. सो फॉर एग्जांपल नेविरैपिन, देन डेलाविरिडिन, देन इट्रावायरिन, rilpavirin. So virin and ridin, rapine, viridin and virin or viridin or rapine. So these are the non-nucleotide reverse transcriptase inhibitor. NRTIs, non-nucleotide reverse transcriptase inhibitor का एग्जांपल क्या है, आप याद रख सकते हो एंडिंग में जो सफिक्स लगा है उसको. सो रैपिन, नेविरैपिन और रैपिन की जितने भी ड्रग्स आएंगे तो वो सब NRTI की कैटेगरी में आएंगे. डेलावेरिडिन, वेरिडिन का, वायरिडिन का जितना भी ड्रग आएगा वो इसके अंदर आएगा. रिपा वायरिन, ठीक है, ये वायरिन भी इसके अंदर आएगा. लेकिन वुडीन का ड्रग जो वुडीन, VUDIN, तो इसमें आएगा न्यूक्लियोटाइड रिवर्स ट्रांसक्रिप्टेज. वीर भी इसमें आएगा, वीर जैसे टेनोफोवीर, अबाकावीर, ठीक है, वो भी इसमें आएगा, वीर एंड वुडीन्स. वीर एंड वुडिन्स आप कॉमनली याद सकते हो, याद रख सकते हो. वीर्स एंड वुडिन्स आर द nucleotide reverse transcriptase inhibitors, and nevirapine, rapine, then viridine are the non-nucleotide reverse transcriptase inhibitor. So they are the inhibitors of reverse transcriptase enzyme. That's why they are effective in case of AIDS or HIV virus. Now next category, drug effective against the same retrovirus. Next category is protease inhibitor. Next one is protease inhibitors. Protease inhibitors. Okay, so protease inhibitors like ritonavir, etanzanavir, then indinavir, nelfinavir, sequinavir, then lopinavir. So these are the protease inhibitor. So remember, protease inhibitor are nothing but the vir, ritonavir, then atazanavir, indinavir, nelfinavir, okay, then sequinavir and lopinavir. These are the protease inhibitor. Now next is the entry inhibitor. So entry inhibitor is there. For example, this is drug N-fluviratide. Okay, N-fluviratide, N-fluviratide. Okay, so that is the entry inhibitor. Now again, this next class is a CCR5 receptor inhibitor. That means chemokinin receptor 5 inhibitors. Chemokinin receptor 5 inhibitors. So again, it will also inhibit the entry only, but it is a specific receptor is mentioned over here. So ये जो मेरा वायरक है, it is also entry inhibitor only, but it will act on this specific receptor. Okay, so CCR5 receptor inhibitor, chemokinin receptor 5 inhibitor, मेरा वायरक, इसके ऊपर भी क्वेश्चन आ जाता है. ठीक है. Now next class is integrase inhibitor. So these are the two drugs, raltegravir and dolutegravir. Raltegravir and dolutegravir, these are the integrase inhibitor. Now integrase, protease, these are the enzymes required for various purposes in the viruses. ठीक है. So वायरस में, वो सेल में उसका मल्टीप्लिकेशन जो होता है, उसका मैचुरेशन जो होता है, ठीक है, उसका इंटीग्रेशन जो होता है, उसका डीएनए और ह्यूमन का डीएनए, सो उसके लिए एंजाइम्स लगते हैं. दीज आर द एंजाइम्स फॉर द रिक्वायर्ड फॉर द various steps in the various steps of virus in the cell, and that's why these are the targets for ड्रग्स. ओके, फॉर एग्जांपल प्रोटीज क्या करेगा, प्रोटीज जैसे ही उसका इनिशियल प्रो प्रोटीन, उसको बोलते हैं प्रो वायरल प्रोटीन. ओके, सो इनिशियल प्रोटीन्स ऑफ वायरल पार्टिकल, सो प्रो वायरल प्रोटीन जो बनता है, सो दैट इज अ इनएक्टिव फॉर्म. सो प्रोटीएज एंजाइम विल ब्रेक डाउन दैट प्रो वायरल प्रोटीन एंड विल गिव यू सम फंक्शनल प्रोटीन. तो उसको नॉन फंक्शनल होता है प्रो वायरल, तो उसको फंक्शनल बनाएगा कौन, प्रोटीज. दैट्स व्हाई प्रोटीज इनहिबिटर्स आर द ड्रग. They will prevent the maturation of virus. They will prevent the assembling of virus. So rotenavir, atazanavir, indinavir, nelfinavir, sequinavir, lopinavir. So then entry inhibitor and CCR5 receptor inhibitor. These two drugs will prevent the entry of virus inside the cell. Okay. Then integrase inhibitor. So integrase means integrase is the enzyme. ठीक है. जैसे उसने पेनिट्रेशन किया सेल में और बाद में आपका उसका खुद का डीएनए रिलीज किया या आरएनए रिलीज किया. ठीक है. नाउ दैट डीएनए आरएनए रिलीज जो है, दैट डीएनए आरएनए गेट्स इंटीग्रेटेड विद द सेल. ठीक है. आपका खुद का भी डीएनए होगा ना सेल में, तो वो सेल के साथ में वायरस का डीएनए इंटीग्रेट होता है, मतलब साथ में बन जाता है. सो दैट इज डन बाय वन एंजाइम नोन एज इंटीग्रेज. Okay, so integrase will prevent the integration of viral DNA and your host DNA. And अगर आप viral DNA और आपका host DNA integrate हो गया, that means your cell is infected, okay, and permanently. So that's why. So integrase inhibitor will prevent the integration of viral DNA plus viral DNA plus host DNA. So reltagravir and dolutegravir, these are the two drugs of integrase inhibitor. Next one is a classification based upon the mechanism of action. So again, it is a type of classification based upon the mechanism of action. So nucleic acid synthesis inhibitors. So those drugs which will inhibit the nucleic acid synthesis, like idoxuridine, then acyclovir, ribavirin, trifluridine, and then azidothymidine. So these are the drugs which will inhibit the nucleic acid synthesis. Then few drugs will inhibit the attachment and penetration of virus. So amantadine will prevent the uncoating and penetration attachment also. Zamna with zanamivir. Amantadine and zanamivir. Amantadine and zanamivir. They will inhibit the penetration and attachment. Reverse transcriptase inhibitor, we have seen examples. For example, lamivudine. Udine जो होते हैं, udines, they are reverse transcriptase inhibitor. then immunomodifiers. So immunity response जो है cell का, that will be modified by obviously antibodies. So interferon, gamma globulins, monoclonal antibodies, they will modify the immune mechanism of your cell, immunity of your cell. Okay, in case of particularly HIV infection. So this was the classification. Now individual antiviral drugs. Now individual anti-herpes drugs. Individual anti-herpes drug. Okay, so individual anti-herpes antiviral drugs. So as the name indicates, anti-herpes means they are only effective against they are mostly effective against the they are mostly effective in the herpes zoster virus. So herpes जो होता है herpes, जिससे होता है ना आपके मुंह के ऊपर एक वायरल इन्फेक्शन होता है, तो दैट इज herpes. आपको नागिन आ गया, नागिन आना मतलब दैट इज ऑल्सो herpes zoster virus. एसाइक्लोविर, फर्स्ट ड्रग इज एसाइक्लोविर. इसके बारे में क्वेश्चन आता है जीपैट में, मैकेनिज्म इसका. So acyclovir, okay. So acyclovir is effective against the herpes simplex virus. This acyclovir is effective against this herpes simplex virus type 1 type 2. दो टाइप होता है उसका herpes simplex का, उसको बोलते हैं herpes simplex virus type 1 type 2. Then it is also effective against the varicella zoster virus. Varicella zoster means this virus is responsible for your measles. वायरसला जोस्टर में आपको कान्नेजा का जो माता जो होता है पेंडेमिक इपिडेमिक ऑफ योर चिकन पॉक्स, सॉरी नॉट मीजल, चिकन पॉक्स. सो वायरसला जोस्टर इज द वायरस रिस्पॉन्सिबल फॉर द इपिडेमिक ऑफ योर चिकन पॉक्स. चिकन पॉक्स आपको हुआ होगा कभी बचपन में या फिर कभी. सो दैट इज बिकॉज ऑफ द वायरसला जोस्टर. सो वायरसला जोस्टर विल कॉज चिकन पॉक्स. सो दिस असाइक्लोविर इज इफेक्टिव इन द herpes simplex virus. It's effective against the varicella zoster virus. Again, it is effective against the Epstein-Barr virus, that is known as EBW, EBV. So, varicella zoster virus means that will cause a chicken pox in you. Chicken pox जो होता है बहुत बड़े-बड़े ये आता है बबल्स आते हैं स्किन के ऊपर और उसमें एक वाटरी फ्लूइड होता है, उसमें वायरस मल्टीप्लाई होता है. ठीक है, और वो बहुत ही तकलीफ देह होता है, बहुत ही इरिटेटिंग होता है और बहुत ही ज़्यादा बर्निंग सेंसेशन होता है उसमें. I think I was suffering from this chicken pox in 2018. I got an infection of varicella zoster in 2018. जिनका immunity कम होता है, so those people or those patients whose immunity is less or whose immunity is not normal, whose immunity is decreased, or any patient which is immunocompromised, so they may get the infection of varicella zoster. Okay, so एक बार अगर varicella zoster का epidemic आ गया ना, तो पूरे मोहल्ले में ये होता है. ठीक है. Now next one is what is the mechanism of action of this? So mechanism of action. So acyclovir will get entry into the infected cells. Okay, then it will be converted into acyclovir triphosphate. Okay, acyclovir will get entry into the cell. Okay, after that it will be converted into acyclovir triphosphate. Remember, acyclovir triphosphate. This now acyclovir triphosphate which will prevent the viral DNA synthesis by inhibiting the viral DNA polymerase and causing DNA chain termination. So ये acyclovir जब सेल में जाएगा तो इसका कन्वर्जन होगा acyclovir triphosphate में. जैसे सेलुलर काइनेज नाम का एक एंजाइम होता है, that cellular kinase will convert acyclovir first into acyclovir diphosphate. Then again it will convert to acyclovir triphosphate. And this acyclovir triphosphate will inhibit the DNA synthesis of virus by inhibiting the DNA polymerase enzyme. कि ये acyclovir triphosphate DNA polymerase enzyme जो है आपका उसको इनहिबिट करेगा ताकि वायरस का प्रोटीन सिंथेसिस ना हो. DNA polymerase मतलब रेप्लिकेशन नहीं होगा, ट्रांसक्रिप्शन नहीं होगा, ट्रांसलेशन नहीं होगा. Okay, so that's how acyclovir will act. What are the adverse drug reaction? So ADRs, common adverse drug reaction like nausea, vomiting, diarrhea. Okay, these are the common nausea, vomiting, diarrhea, NVD. Then headache, occasionally it will cause headache. When used topically, it will cause burning and irritation. And when acyclovir is used intravenously, it can cause renal and neurotoxicity, which is uncommon. इतना common नहीं है. Occasionally ये हो सकता है IV देने के बाद. लेकिन if you use topically, it will cause burning and irritation. So no major adverse effects are there if you used orally. Orally अगर देंगे तो उतना adverse effect नहीं है, सिर्फ nausea, vomiting, diarrhea, occasionally headache. Okay. Now, what are the uses of this acyclovir? Uses of this acyclovir: HSV infection, herpes simplex virus infection, type 1 and type 2. जो भी टाइप 1 टाइप 2 herpes simplex virus का उसका इन्फेक्शन है, जैसे आपके जो पर्टिकुलरली जो होता है ये मोस्ट कॉमन herpes simplex virus इज देयर इन केस ऑफ HIV पेशेंट, जो जिनको HIV हुए तो उनका बहुत सारा मतलब मुंह में छाले रहना या मुंह के अराउंड मुंह के जो वट एवर द म्यूकस लेयर इन देयर इज माउथ, सो देयर विल बी अल्सरेशन, so herpes simplex virus spotting will be there outside the, जैसे आपका होता है ना, बोलते हो मेरा मुंह मुंह होता है, वो बोल नहीं सकता मैं, उसका नाम नहीं याद है. So that is herpes simplex virus. So it is used in the treatment of herpes simplex virus infection of mouth, face, skin, okay, and brain. Then type 2 herpes simplex virus mostly causes the infection of genital organs, rectum, skin, and meninges. Okay, meninges. So encephalitis जिसको बोलते हैं अपन, herpes simplex virus का encephalitis, meningitis. Type 1 herpes simplex will mostly infect your mouth, face, skin, and brain. Okay. Type 2 herpes simplex virus mostly infect your genital organs, जैसे आपका मेल्स या फीमेल्स में rectum, then skin and meninges. IV intravenous acyclovir is a drug of choice in case of herpes simplex virus encephalitis. Remember, IV acyclovir is a drug of choice for herpes simplex virus encephalitis and herpes simplex virus केराटोकंजक्टिवाइटिस. ठीक है, तो herpes simplex virus का अगर आपको आई में इन्फेक्शन हो गया और आपका अगर ब्रेन में इन्फेक्शन हो गया, सो ये एन्सेफेलाइटिस हो गया या फिर केराटोकंजक्टिवाइटिस हो गया, सो drug of choice is acyclovir. There is another brother of this acyclovir, another brother from brother from another mother. Now another brother of acyclovir is valacyclovir. Valacyclovir. This valacyclovir is the prodrug of acyclovir. So acyclovir, valacyclovir is the prodrug of acyclovir, means after giving the valacyclovir it will be converted into acyclovir in the body. Okay. Then famciclovir. Famciclovir is again a product of peniciclovir. 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 Famciclovir. Okay. So famciclovir is a product of peniciclovir. So yeah, all these drugs are related with the acyclovir. Rest of the things are similar with the acyclovir. बाकी का जो चीजें है जैसे mechanism, adverse effect similar to acyclovir. Next one is a ganciclovir. Sorry. Next one is a ganciclovir. और एक drug है ganciclovir. Okay, ganciclovir. Ganciclovir is effective against the herpes simplex virus. Particularly, it is effective against the cytomegalovirus. Okay, so against the cytomegalovirus, ganciclovir is used. अगर साइटोमेगलोवायरस का इन्फेक्शन है तो ganciclovir यूज करते हैं, अगर herpes simplex का है तो acyclovir यूज करते हैं. And it is used in the immunocompromised patient with cytomegalovirus infection of retinitis. It is a drug of choice in case of cytomegalovirus infection of retina or CMV retinitis. Okay, so drug of choice in case of CMV retinitis is a ganciclovir. Remember, आप लिख लो या देखो. Then next one is idoxuridine. Next one is idoxuridine. Next one is idoxuridine. Now it is again effective in case of DNA virus only. Okay, it will inhibit the DNA synthesis, okay, of virus. Again, idoxuridine is a effective in DNA virus only, and it will inhibit the DNA synthesis of virus. And this drug is used topically because systemic use of this idoxuridine is too toxic. So that's why it is used topically. In case of herpes simplex keratitis, trifluridine is not used topically for herpes simplex eye infection. Trifluridine is a derivative of idoxuridine. Idoxuridine का ही trifluridine derivative होता है. So that is used in case of herpes simplex virus eye infection. But this drug is only used topically, not for systemic use. Now next one is foscarnet. Foscarnet is used to treat the cytomegalovirus retinitis, alternative to ganciclovir. First drug of choice in CMV retinitis is a ganciclovir, and if it is not effective, then only foscarnet is used. Okay, so it is alternative. Fomiviracin is an alternative drug to the other drugs. Alternative to the other drug when they are ineffective in case of cytomegalovirus retinitis. मतलब अगर आपका गैंसाइक्लोविर भी इफेक्टिव नहीं है, फॉस्कारनेट भी इफेक्टिव नहीं है, then you have to use fomiviracin. So alternative drug to the other when they are ineffective in case of cytomegalovirus CMV retinitis. ये रेटिना जो आपका आइज का रेटिना है उसको इन्फेक्ट होता है साइटोमेगालोवायरस, तो उसको बोलते हैं साइटोमेगालो CMV retinitis. Okay. Next one is cidofovir. Cidofovir. So it is a broad spectrum antiviral drug and it again acts by inhibiting the DNA virus, viral DNA synthesis. Then effective against the herpes zoster. It is effective against the herpes virus like herpes zoster virus, cytomegalovirus, Epstein-Barr virus, human papilloma virus, then adenovirus. Okay. Then it is used intravenously for prevention and progress of cytomegalovirus retinitis in the HIV patient. Then it is also topically used. It is also used topically for HPV, human papilloma virus skin infection. HPV means human papilloma virus infection. Okay. Now we'll see again the mechanism of your acyclovir. The mechanism acyclovir. Acyclovir कैसे? So acyclovir. यहाँ पे देखो acyclovir. Acyclovir will be converted into acyclovir monophosphate. Acyclovir monophosphate. Acyclovir will get entry into a cell. It will be converted into acyclovir monophosphate by viral kinase enzyme. वायरस में kinase enzyme मतलब वो convert करेगा acyclovir को monophosphate में. Okay. Then acyclovir monophosphate is converted into acyclovir diphosphate by cellular kinase. मतलब ये वायरस का एंजाइम है, वायरल काइनेज, और ये आपका होस्ट सेल का एंजाइम है, सेलुलर काइनेज. ठीक है. तो सेलुलर काइनेज विल कन्वर्ट acyclovir monophosphate into acyclovir diphosphate. Again acyclovir diphosphate will be converted into acyclovir triphosphate by cellular kinase. Cellular kinase will convert acyclovir diphosphate, acyclovir monophosphate first to diphosphate and then triphosphate. Now this acyclovir triphosphate inhibit the DNA polymerase, viral DNA polymerase. Okay, it inhibit the viral DNA polymerase. Again, this also cause the prevent the viral DNA chain elongation, or it causes viral DNA chain termination. So this is the mechanism of action of acyclovir. Next class of drug is nothing but the drug effective against the influenza or anti-influenza virus drug. नेक्स्ट क्लास इज एंटी इन्फ्लुएंजा वायरस ड्रग. सो इसमें जो भी एग्जांपल है वो देखेंगे वन बाय वन. सो इट इज जस्ट समरी, इट इज जस्ट एसेंस ऑफ योर एंटीवायरल ड्रग्स, कि हर एक बुक में से निकाल के सब कुछ यहाँ पे रेडीमेड आपको परोस के रखा है, आपको सिर्फ खाना है और पचाना है. सो एंटी इन्फ्लुएंजा वायरस ड्रग और एंटी इन्फ्लुएंजा एंटीवायरल ड्रग. सो फर्स्ट वन इज amantadine and rimantadine. So amantadine and rimantadine, these are the drug, they are mostly active against the influenza virus. So they act by inhibiting the replication of influenza A virus. They also prevent the attachment of virus to the host cell. They also prevent the uncoating of virus to the uncoating of viral DNA. याद रखो, they act by inhibiting the replication of virus, influenza A virus. Also prevent the attachment, also prevent the uncoating of virus. So adverse drug reaction includes nausea, vomiting, diarrhea, then insomnia, ankle edema, etc. Amantadine. Okay. Rimantadine is a derivative of amantadine. Rimantadine is a derivative of amantadine, which is longer acting than amantadine and will have less adverse effect than amantadine. Rimantadine is a longer acting derivative of amantadine and will have less adverse effect than the amantadine. Okay. Now what are the uses of these two drugs? Amantadine and rimantadine का use क्या होता है, ठीक है? So they are used in the prophylaxis of influenza A virus. So they are used in the prophylaxis of influenza A virus. Prophylaxis of influenza A virus. Prophylaxis means to prevent the influenza infection in the patient who have high risk. जिनका immunity कम है, the patient high risk patient means क्या? Suppose that if any patient is diabetic, if any patient is patient has a transplanted his kidney or anything stent, जैसे होता है ना कोरोना वायरस में, the geriatric patients are most prone to infection. Then patient whose immunity is compromised, they are also prone to coronavirus infection, because coronavirus is nothing but it is a type of an influenza virus. It is type of influenza virus. Okay, so This amantadine is useful in the prophylaxis or prevention of influenza A. Again, it will reduce the duration of disease and severity of disease in case of epidemic. Epidemic मतलब क्या? आप जब चल रहा है कोरोना का पेंडेमिक. Pandemic is nothing but when a disease spreads all over the world. Okay, that is known as a pandemic. If a disease is localized particularly in a particular state or particular state or particular country, then it is known as epidemic. ओके, जैसे कोरोना वायरस अगर सिर्फ पहले का था सिर्फ चाइना में था, वहां पे चाइना वाले क्या बोलेंगे उसको, कोरोना वायरस इपिडेमिक. जब वो कोरोना वायरस सब पूरे पूरे वर्ल्ड में घूम के आ गए, मतलब स्प्रेड हो गए, तो उसको बोलेंगे pandemic. Okay, epidemic, pandemic. Okay, so this amantadine rimonabant is they are used to prevent the severity of disease जो है इन्फ्लुएंजा ए का वायरस का जो कॉम्प्लिकेशन है उसको कम करेगा, सीवियरिटी कम करेगा. ओके, एंड ड्यूरेशन विल बी इंक्रीज. अगेन दिस amantadine releases the amantadine जो है it will release the dopamine from nerve endings. Amantadine will release the dopamine from nerve ending, and that's why it is useful in the Parkinson disease also. Okay, this amantadine they are used to rimonabant they are used to prophylaxis of influenza A virus, and the amantadine is also a dopamine releaser. That's why it is used in the treatment of Parkinsonism also. Okay. Now next one is oseltamivir, oseltamivir and zanamivir. Oseltamivir and zanamivir. These are the related drugs. Oseltamivir, you might have heard this name oseltamivir. You might have seen one tablet known as, or you might have heard one tablet known as Tamiflu. So I don't know whether in your state there was a infection of this. You know swine flu. So swine flu was the infection in 2011. Particularly, it was spreading in the Maharashtra region, particularly in Pune region. When I was in a B.Pharm final year, there was a epidemic of your swine flu. Like bird flu is there, swine flu is there. Okay, so both bird flu and swine flu. Bird flu occurs in the uh, birds only, chickens, ducks, and all that. Okay, but swine flu occurs. Swine flu is spread by your pigs. Okay, swine flu is spread by pigs, and bird flu is spread by that birds. Okay, so both in case of both bird flu and swine flu, also oseltamivir is a drug of choice. Oseltamivir is a drug of choice in case of bird flu as well as swine flu. Okay, it is caused by influenza virus only. Okay, now oseltamivir and zanamivir. So they act by inhibiting. They act by inhibiting the enzyme neuraminidase activity, which is essential for release of daughter viron. जो मैंने आपको पहले ही दिखाया था, whenever I was demonstrating you the site of action. So at that time I have told you after the assembly of virus, when maturation of virus is there, it is released from the cell into outside to infect the other cell. So that to release the newly daughter viral particle, neuraminidase enzyme is responsible. It is important. So these drugs will inhibit the neuraminidase enzyme. Okay, oseltamivir and zanamivir, they will inhibit the neuraminidase activity, which is essential for release of daughter viron from the cell. Okay, so oseltamivir, its brand name is Tamiflu, and it is a drug of choice in case of your bird flu and swine flu. Oseltamivir, Tamiflu is a drug of choice used orally for treatment of bird flu and swine flu. It will have nausea, vomiting, diarrhea, like adverse effect. Other influenza infection, bird flu, swine flu, and other influenza infection. Oseltamivir is a drug of choice. Okay? Then zanamivir is a drug given by inhalation and can cause respiratory distress. So this आपका जो oseltamivir है, that is given by oral route. Zanamivir is given by inhalation route. Next drug is a docosanol. It inhibits the viral entry. It inhibits the viral entry into the host cell and suppresses the viral replication also. And it is topically used for herpes infection. So they can ask around one or two questions from this antiviral chapter. Now other antiviral drugs. So other antiviral drug. So uncategorized. For example, ribavirin. So it is a broad spectrum antiviral drug. क्या है ये? ब्रॉड स्पेक्ट्रम है, ब्रॉड स्पेक्ट्रम एंटीवायरल ड्रग है। सो इट इज आगे इफेक्टिव अगेंस्ट ब्रॉड स्पेक्ट्रम, मतलब वो उसका स्पेक्ट्रम ऑफ एक्टिविटी ज्यादा हुआ है ना, जैसे दैट विल इनहिबिट डीएनए वायरस also as well as RNA virus. So ribavirin is a broad spectrum antiviral drug against the DNA as well as RNA virus. Influenza A virus. Then it is also active against the respiratory syncytial virus. That's why it is a broad spectrum antiviral drug. So it is effective against the broad spectrum antiviral drug, effective against the most of the DNA and RNA virus, effective against the influenza A virus, respiratory syncytial virus, and this drug is used in the treatment of RST bronchiolitis, रेस्पिरेटरी सिंसिटिव वायरस की वजह से जो इन्फेक्शन होता है ब्रोंकियस में उसको बोलते हैं आरएसवी ब्रोंकियोटाइटिस। ओके, सो इट इज यूज्ड इन द आरएसवी ब्रोंकियोटाइटिस in case of children's, and it is also used in the prophylaxis of influenza. Okay, then also used in the prophylaxis of measles in case of immunocompromised patient. Then next one is the interferon. Next is the interferon. So interferon, they are nothing but the antibodies against the virus. जैसे आपका जो है आपके बॉडी में अगर कोई इन्फेक्शन आ रहा है, जैसे बैक्टीरियल इन्फेक्शन आ गया, तो उसके अगेंस्ट में आपकी बॉडी, आपका जो इम्यूनिटी सिस्टम है उसके अगेंस्ट में आपकी बॉडी एंटीबॉडी बनाती है। ठीक है, सो अगेंस्ट द एंटीजन योर body or immunity system will produce antibodies. Okay, so likewise that against the viral infection your body will produce interferons. Okay, so interferons, so antibody against the virus, that is interferons. So antibody, particularly they are the cytokinins produced against the infection by the host cell. Okay, so antibody or antibody cytokinin produced against the virus by the host cell. Okay, so that is nothing but the interferon. In the humans there are three type of interferon. First one is alpha. Okay, alpha, beta and gamma. Alpha, beta and gamma interferons are there. So alpha interferon will have 166 amino acid. It will have molecular weight of 1600 to 26 16,000 to 26,000 molecular weight उसका रहेगा alpha interferon का. Then beta interferon and then gamma interferon. So gamma interferon will have 146 amino acid sequence and molecular weight is 20 to 25,000 delta. So that is a interferon, alpha, beta, gamma, three types. They inhibit the replication of many DNA and RNA virus and inhibition of several steps of viral replication or viral reproduction. Now ये क्या है, antibodies है, they will inhibit the replication of DNA virus, RNA virus. They will prevent the different steps like attachment, budding, then uncoating. So they act at a multiple sites. They act at a multiple sites of virus. So interferon, so they inhibit the multiplication of many DNA RNA virus, inhibition of several steps of viral replication like penetration, uncoating, synthesis of mRNA and proteins. So they are again a broad spectrum. Okay, they will have a, because ये specific होता है against में, virus के against में. Okay, and that's why initially when there was a coronavirus pandemic, so our healthcare workers have started plasma, donating the plasma. जिसको हो चुका है ऑलरेडी कोरोना वायरस, उसने क्या करना है, रिकवर होने के बाद प्लाज्मा डोनेट करना है. Why? Because that plasma consists of interferons. That plasma consists of antibodies against the viral infection. Okay, so that is used to prevent the disease for the prophylaxis. So, but that was not much effective because antibodies against the coronavirus will last only for few days or few months. Okay, so adverse effect of interferons. They have a serious adverse effect like neurotoxicity, alopecia, alopecia means loss of hair, neurotoxicity, then arthralgia, headache, hypotension, arrhythmia, and myelosuppression. So these are the adverse effect of your interferons. Now what are the uses of interferon? So they are used in the different conditions. Okay, for example, they are used in the hepatitis B and C. Then Kaposi sarcoma, it is a sarcoma. It is a cancer. Kaposi sarcoma is cancer in the AIDS patient. Then genital warts. ये आपके private part पे कई warts वगैरह हो चुके हैं. So that in case of that interferon is viral warts due to viral infection, due to HPV, human papilloma virus, cell leukemia, then herpes simplex virus infection, herpes zoster, cytomegalovirus. Okay, infection of these three in case of immunocompromised patient. Alpha interferon is used internally for prophylaxis of rhinovirus cold. Okay, so interferon. Now next one is a drug used in the hepatitis.